Hello dear students, welcome to this program. Today we are discussing an interesting topic which is microorganisms growth in food, extrinsic and intrinsic factors, microorganisms causing food spoilage in fresh food, milk and canned food, preparation of fermented food products, fermented milk such as yogurt, curd and cheese and other fermented foods like pickles. Before discussing them in detail, let us first know its objectives. The main objectives are to understand, to know about growth of microorganisms in food and various factors involved, brief description about food spoilage, and preparation of fermented foods. Now let us start with microorganism growth in food. Foods, because they provide nutrients for us, also are excellent environments for the growth of microorganisms. Microbial growth is controlled by factors related to the food itself or intrinsic factors, and also to the environment where the food is being stored, or what are described as extrinsic factors. Now let us know about intrinsic factors in detail. It comprises of food composition. Food composition is a critical intrinsic factor that influences microbial growth. If a food consists primarily of carbohydrates, spoilage does not result in major odors. Thus, foods such as breads, jams, and some fruits first show spoilage by fungal growth. In contrast, when foods contain large amounts of proteins or fats, for example, meat and butter, spoilage can produce a variety of foul odors. One only need think of rotting eggs. Now coming to pH. The pH of a food also is critical because a low pH favors the growth of yeast and molds. In neutral or alkaline pH, foods such as meats bacteria are more dominant in spoilage and putrefaction. Depending on the major substrate present in a food, different types of spoilage may occur. Now, presence and availability of water. The presence and availability of water also affect the ability of microorganisms to colonize foods. Simply by drying a food, one can control or eliminate spoilage processes. Water, even if present, can be made less available by adding solutes such as sugar and salt. Water availability is measured in terms of water activity, denoted by AW. This represents the ratio of relative humidity of the air over a test solution compared with that of distilled water. When large quantities of salt or sugar are added to food, most microorganisms are dehydrated by the hypertonic conditions and cannot grow. Even under these adverse conditions, osmophilic and xerophilic microorganisms may spoil food. Now coming to oxidation reduction potential. The oxidation reduction potential of a food also influences spoilage. When meat products, especially broths, are cooked, they often have lower oxidation reduction potentials. These products, with their readily available amino acids, peptides and growth factors, are ideal media for the growth of anaerobes, including Clostridium. Now coming to physical structure of food. The physical structure of a food can also affect the course and extent of spoilage. The grinding and mixing of foods such as sausage and hamburger not only increase the food surface area and alter cellular structure but also distribute contaminating microorganisms throughout the food. This can result in rapid spoilage if such foods are stored improperly. Vegetables and fruits have outer skin, that is, peels and rinds that protect them from spoilage. Often, spoilage microorganisms have specialized enzymes that help them weaken and penetrate protective peels and rinds, especially after the fruits and vegetables have been bruised. Many foods contain natural antimicrobial substances, including complex chemical inhibitors and enzymes. Coumarins found in fruits and vegetables exhibit antimicrobial activity. Cow's milk and eggs also contain antimicrobial substances. Eggs are rich in the enzyme lysozyme that can lyse the cell walls of contaminating gram-positive bacteria. Now coming to extrinsic factors. It comprises of temperature and relative humidity are important extrinsic factors in determining whether a food will spoil. Relative humidity. At high relative humidities, microbial growth is initiated more rapidly even at low temperatures, especially when refrigerators are not maintained in a defrosted state. When dry foods are placed in moist environments, moisture absorption can occur on the food surface, eventually allowing microbial growth. Atmosphere. The atmosphere in which the food is stored also is important. This is especially true with shrink-packed foods because many plastic films allow oxygen diffusion, which result in increased growth of surface-associated microorganisms. Excess carbon dioxide can decrease the solution pH, inhibiting microbial growth. Storing meat in a high carbon dioxide atmosphere inhibit gram-negative bacteria, resulting in a population dominated by the lactobacilli. The observation that food storage atmosphere is important has led to development of modified atmosphere packaging, that is MAP. By the use of modern shrink-wrap materials and vacuum technology, it's possible to package foods with controlled atmosphere. Now coming to microbial growth and food spoilage. Because foods are such excellent sources of nutrients, if the intrinsic and extrinsic conditions are appropriate, microorganisms grow rapidly and make what once was an attractive and appealing food into sour, foul-smelling or fungus-covered mass suitable only for the garbage can. Microbial growth in foods can lead to visible changes, including a variety of colors caused by spoilage organisms, which often have been associated with miracles and witchcraft. One of the most famous is a report of blood on communion wafers and other bread called the Miracle of Bolsena, which occurred in 1263. The riddle was eventually solved by Bartolomeo Bizia in 1879. He described the bacterium responsible for this phenomena. He also named the bacterium Serratia marcescens. Now coming to spoilage of meat and dairy products. Meat and dairy products with their high nutritional value and the presence of easily usable carbohydrates, fats and proteins provide ideal environments for microbial spoilage. Proteolysis and protofaction are typical results of microbial spoilage of such high-protein materials. Unpasteurized milk undergoes a predictable four-step succession during spoilage. Acid production by Lactococcus lactis is followed by additional acid production associated with the growth of more acid-tolerant organisms such as Lactobacillus. At this point, yeast and molds become dominant and degrade the accumulated lactic acid, and the acidity gradually decreases. Eventually, protein-digesting bacteria become active, resulting in a putrid odor and bitter flavor. The milk, originally opaque, can eventually become clear. Now coming to spoilage of fruits and vegetables. Vegetables. The main source of microorganisms in vegetables are soil, water, air and other environmental sources and can include some plant pathogens. Fresh vegetables are fairly rich in carbohydrates, 5% or more, and low in proteins, about 1 to 2%, and except for tomatoes, have high pH. Microorganisms grow more rapidly in damaged or cut vegetables. The presence of air, high humidity, and high temperature during storage increases the chances of spoilage. The common spoilage defects are caused by molds belonging to genera Penicillium, Phytophthora, Alternaria, Botaris, and Asparagus. Microbial vegetable spoilage is generally described by the common term rot, along with the changes in the appearance such as black rot, grey rot, pink rot, soft rot, and stem end rot. Now coming to fruits. Fresh fruits have high carbohydrate content, 10% or more, very low protein, that is 10%, but have pH 4.5 or below. Thus, microbial spoilage of fruits and fruit products is confined to molds, yeast, and acidic bacteria like lactic acid bacteria, Acetobacter, Glyconobacter. Like fresh vegetables, fresh fruits are susceptible to rot by different types of molds. from genera Penicillium, Asparagus, Alternaria, Botatus, Rhizopus, and others. According to the changes in appearance, the molds spoilage are designated as black rot, grey rot, soft rot, brown rot, and others. Bacterial spoilage associated with the souring of berries and figs have been attributed to the growth of lactic acid and acetic acid bacteria. To reduce spoilage, fruits and fruit products are preserved by refrigeration, freezing, drying, and reducing AW, that is water potential, vacuum packaging, and heat treatment. Now coming to spoilage of fish. Fish have high nitrogen content but no carbohydrate. The microbial quality of fish, and especially shellfish, is heavily influenced by the quality of the water from which they are harvested. Unsanitized processing steps are principal culprits in fish products with high microbial loads. In general, frozen fish products have lower counts than fresh products. Bacteria on fresh fish are concentrated on the outer slime, gills, and intestine. Furthermore, algal toxins contaminate fish and thus affect the health of marine animals higher in the food chain. They also can contaminate shellfish and finfish, which are later consumed by humans. Most toxins are produced by dinoflagellates, but some diatoms also are toxic. Major human diseases that result from algal toxins in marine products include amnesic, diuretic, and neurotoxic shellfish poisoning. These complex algal toxins, most of which are temperature stable, are known to cause peripheral neurological system effects, often in less than 1 hour after ingestion. Now coming to spoilage of canned foods. Canned foods can undergo spoilage despite safety precautions. Spoilage can be due to spoilage prior to canning, underprocessing during canning, or leakage of contaminated water through cane seams during cooling. Canned foods are heat treated to kill microorganisms present, and the extent of heat treatment is predominantly dependent on the pH of the food. High pH foods are heated to destroy most heat-resistant spores of pathogenic bacteria, that is Clostridium botulinum, to ensure that a product is free of any pathogen. However, spores of some spoilage bacteria which have greater heat resistance can survive. Such products are called commercially sterile foods. The other food group designated as a low pH or high acid food with pH 4.6 or below is given heat treatment to kill all vegetative cells and some spores. Although low pH will inhibit germination and growth of Clostridium botulinum, spores of some acidic thermophilic spoilage bacteria can germinate and grow when the products are stored at higher temperatures, even for a short time, which facilitates germination. Some spores of thermoduric mesophilic spoilage bacteria, including pathogenic, can also survive heating in these products, but they are inhibited by the low pH. Canned food spoilage is due to non-microbial, that is chemical and enzymatic reactions, and microbial reasons. Production of hydrogen, that is hydrogen swell, carbon dioxide, browning, corrosion of canes due to chemical reactions, and liquefaction, gelination, discoloration of products due to enzymatic reactions are some examples of non-microbial spoilage. Microbial spoilage is due to three main reasons: inadequate cooling after heating or high temperature storage allowing germination and growth of thermophilic spore formers, inadequate heating resulting in survival and growth of mesophilic microorganisms, and leakage in the canes allowing microbial contamination from outside following heat treatment and the growth. Now coming to microbiology of fermented foods. Over the last several thousand years, fermentation has been a major way of preserving food. Microbial growth, either of natural or inoculated populations, causes chemical and textural changes to form a product that can be stored for extended periods. The fermentation process also is used to create new, pleasing food flavors and odors. The major fermentations used in food microbiology are lactic, propanoic, and ethanolic fermentations. These fermentations are carried out with a wide range of cultures, many of which have not been characterized. Now coming to fermented milk. Throughout the world, at least 400 different fermented milks are produced. These fermentations are carried out by Mesophilic, Thermophilic and Therapeutic lactic acid bacteria as well as by yeast and molds. Major examples of these fermentation types are Mesophilic milk fermentation. Mesophilic milk fermentation results from similar manufacturing techniques in which acid produced through microbial activity causes protein denaturation. To carry out the process, one usually inoculates milk with the desired starter culture, incubates it at optimum temperature approximately 20 to 30 degrees Celsius and then stops microbial growth by cooling. Lactobacillus species and Lactococcus lactis cultures are used for aroma and acid production. The organism Lactococcus lactis converts milk citrate to diacetyl, which gives a special buttery flavor to the finished product. The use of these microorganisms with skim milk produce cultural buttermilk, and when cream is used, sour cream is the result. Now coming to thermophilic milk fermentation. In addition to mesophilic milk fermentations, thermophilic fermentations can be carried out at temperatures around 45 degrees Celsius. An important example is yogurt production. Yogurt is made using a special starter culture in which two major bacteria are present in 1:1 ratio, Streptococcus thermophilus and Lactobacillus bulgaricus. With these organisms growing in concert, acid is produced by Streptococcus and aroma components are formed by the Lactobacillus. Freshly prepared yogurt contains 10⁻⁹ bacteria per gram. Now coming to yeast lactic fermentation. Yeast lactic fermentations include kefir, a product with an ethanol concentration of up to 2%. This unique fermented milk originated in the Caucasus Mountains and it is produced east into Mongolia. Kefir products tend to be foamy and frothy due to active carbon dioxide production. This process is based on the use of kefir grains as an inoculum. These are coagulated lumps of casein that contains yeast, lactic acid bacteria and acetic acid bacteria. In this fermentation, the grains are used to inoculate the fresh milk and then recovered at the end of the fermentation. Now coming to mold lactic fermentation. Mold lactic fermentation results in a unique finished fermented milk called villi. The milk is placed in a cup and inoculated with a mixture of the fungus Geotrichum candidum and lactic acid bacteria. The cream rises to the surface and after incubation at 18 to 20 degrees Celsius for 24 hours, lactic acid reaches a concentration of 0.9%. The fungus forms a velvety layer across the top of the final product, which also can be made with a bottom fruit layer to add extra flavor. Now coming to cheese production. Cheese is one of the oldest human foods and is thought to have been developed approximately 8,000 years ago. About 2,000 distinct varieties of cheese are produced throughout the world, representing approximately 20 general types. Often cheese are classified based on texture or hardness as soft cheese like we have cottage or cream and berry, semi-soft cheese like Münster, Limburger, Blue, or hard cheese like Cheddar, Colby and Swiss, or very hard cheese like Parmesan. All cheese result from a lactic acid fermentation of milk, which result in coagulation of milk proteins and formation of a curd. Rennin, an enzyme from calf stomachs but now produced by genetically engineered microorganisms, can also be used to promote curd formation. After the curd is formed, it is heated and pressed to remove the watery part of the milk or whey, salted and then usually ripened. The cheese curd can be packaged for ripening with or without additional microorganisms. Cheese curd inoculation is used in the manufacture of Yogofold and blue cheese. In this case, Penicillium yogoforti spores are added to the curd just before the final cheese processing. Sometimes the surface of an already formed cheese is inoculated at the start of ripening. For example, Camembert cheese is inoculated with spores of Penicillium camemberti. The final hardness of the cheese is partially a function of the length of ripening. Soft cheese are ripened for only about 1 to 5 months, whereas hard cheese need 3 to 12 months, and very hard cheese like Parmesan require 12 to 16 months ripening. The ripening process also is critical for Swiss cheese. Gas production by propanoic bacterium contributes to final flavor development and hole or eye formation in the cheese. Some cheese are soaked in brine to stimulate the development of specific fungi and bacteria. Limburger is one such cheese. Now coming to other fermented foods. Many other plant products can be fermented. These include tofu, which is produced by the fermentation of tofu, a chemically coagulated soybean milk product. To carry out the fermentation, the tofu curd is cut into small chunks and dipped into a solution of salt and citric acid. After the cubes are heated to pasteurize the surfaces, Actinomycor elegans and other Micro species are added. When a white mycelium develops, the cubes, now called fetus, are aged in salted rice wine. This product has achieved the status of a delicacy in many parts of the Western world. Another popular product is tempeh, a soybean mash fermented by Rhizopus. Sauerkraut or sour cabbage is produced from wilted shredded cabbage. Usually the mixed microbial community of the cabbage is used. A concentration of 2.2 to 2.8% sodium chloride restricts the growth of gram-negative bacteria while favoring the development of the lactic acid bacteria. The primary microorganisms contributing to this product are Lictonostoc mesenteriodis and Lactobacillus platinum. A predictable microbial succession occurs in sauerkraut development. The activities of the lactic acid producing cocci usually cease when the acid content reached 0.7 to 1.0%. At this point, Lactobacillus plantarum and Lactobacillus brevis continue to function. The final acidity is generally 1.6 to 1.8, with lactic acid comprising 1.0 to 1.3% of the total acid in a satisfactory product. Now coming to last one, that is pickles. Pickles are produced by placing cucumbers and such components as dill seeds in casks filled with a brine. The sodium chloride concentration begins at 5% and rises to about 16% in 6 to 9 weeks. The salt not only inhibits the growth of undesirable bacteria but also extracts water and water-soluble constituents from the cucumbers. These soluble carbohydrates are converted to lactic acid. The fermentation, which can require 10 to 12 days, involves the development of Lictonostoc mesenteriodis, Enterococcus facilis, Pediococcus cerevisiae, Lactobacillus brevis, and Lactococcus plantarum. Lactococcus plantarum plays a dominant role in this fermentation process. Sometimes, to achieve more uniform pickle quality, natural microorganisms are first destroyed and the cucumbers are fermented using pure cultures of Plantarum cerevisiae and Lactobacillus plantarum. That's about this topic. Hope you enjoyed and understood. Thank you.